Professor Izumi Ohzawa will give us talk. He's working in Osaka University, and he is, of course, a neuroscientist, and he's also very strong to computational algorithm or something. So please, okay.
Hello, everyone. Thank you for having me here. This talk is maybe a little bit weird. Recently, there have been a lot of computational models, neural net models, and I finally decided that I have to pay a close attention to the results because they're working so well. And so the thing that I noticed about these networks is that starting with neocognitron, I asked Dr. Fukushima as well, nearly all neural nets for vision have a cascading hierarchical structure consisting of simple, complex structure, repeating, filtering, and then non-linearity, and then filtering and pooling. Maybe along with normalization there. So that's the N is just repetition, okay? So then I started looking at those things, and I started asking these strange questions, like, do we really need complex cells? And no, I'm not really disputing anything that Hubel-Wiesel discovered. I mean, in V1, it's clear we have them, but I'm not sure if we really need them in the higher order areas. And for example, I mean, is empty cell simple or complex? And maybe you can mull on that question a little bit. And then I started asking whether there's a difference between filtering and pooling, really. And so I tried to sort that out. And so I don't have all the answers to these questions. And I need maybe your help. And that's the reason that I'm here. Okay, so this is Fukushima, the neocognitron. And here you have the input, the retina, and then simple cell layer and a complex cell layer. This is V1. And then, simple cell, complex cell, simple cell, complex, simple complex, and so on. So repeating cascading structure that I talked about. And it's happening in other models as well. Here, it's a stereo model, and the image is here. And then the V1 convolution kernels, and then they do subsampling. And I think it's the pooling is needed for anti-aliasing, and then decimation or subsampling. So that's how I understand it. And then convolution happens again. This is a simple cell layer in a way, and then complex subsampling again, and so on. And then from the latest work by Dr. DiCarlo, I mean, here he has convolution input into the first linear, non-linearity and pooling, and these are repeated. LN stands for this box, as I understand it. So this must be the simple cell, and then the pooling probably is complex, and then with normalization and so on. So these are repeated structures, like in other work. So, but as a physiologist, we have an electrode into somewhere here, and my question is, where is my cell? I mean, in this structure. I mean, it's often misunderstood in the neural network community that when you jump from one layer to another, all the cells change their property, like a new property develops, but that's not entirely true. Like empty cell, I mean, there are cells that are pattern direction selective that detect the velocity of a field, but then there are empty cells that respond just like V1 complex cells. So the things are a little grayish there. So, and even in V1, it's not just straight simple complex cells. There are cells that have higher order attributes, like centers around so-called end stopping, or surround suppression. So surround suppression probably will be cells about here. And so I tried to summarize the features of these convolution filtering and then pooling. And these are sort of my tentative results. And the red indicates somewhere that are different between filters and pooling. Let's go through them. Filters, it's a linear sum of outputs of multiple cells in the previous stage. And then the difference is that in these connection weights to the previous layer, positive and negative connection weights are allowed. And that's probably not true for pooling. Pooling only pools with positive weights and nothing negative. Okay, and then rectification, that happens, that stands for nonlinearity, but that comes automatically if you have a cell. Cell, when you hyperpolarize, doesn't do anything, it doesn't fire action potentials. So that's really not a decisive factor. And these things tend to be simple cells, and these things tend to be complex cells if you do pooling. And so, I mean, new tuning, like a new selectivity like orientation tuning and velocity tuning probably develops with filters. And people tend to think, at least we tend to think that it's kind of boring, pooling. So nobody studies pooling, but we do. We have been doing studies of pooling. So maybe I'm probably qualified to talk about this. So I'm here today. So here, I mean, this is probably a strange question, but I mean, I have to think about it. I mean, this is the standard energy model of a complex cell organization. So here you have 4 simple cells with, a 90-degree step phases, and then it just sums those after nonlinearity, half-squaring nonlinearity. So this is a complex cell, and then the higher stage will collect from these complex cells and organize a receptive field like this. And this will give you something like a surround suppression. Okay, but if you think about it, I mean, if you have a cell here, I mean, can't you just connect these things directly over here? Right, I mean, it doesn't really do any difference. And so whether you have an intermediate summing stage, and I mean, it's bracketed, but if you unwrap those brackets, then do the same thing for all these things directly from simple cell. That'll work the same way. And so I don't think my sort of question is really so out of place here. So only reason that I can think of is maybe you have to say long distance wires from one area to next. In other words, I mean, here you have, with a minimal model, you need 4 wires directly to higher order areas, but if you sum it up here once, and then you only need one wire. And this 4 is really minimal, and I think there's a lot more of simple to complex convergence. So, one thing that I also noticed was that this is the reason that V1 complex cells exist. In other words, if you have a simple cell and like this, Gabor-like receptive field, you have on and off subregion. And if you need to detect contrast, regardless of where the bright bar lies as opposed to dark bar lies, if you don't care about the exact position, but just care about whether there is a contrast, you want to do this kind of computation. You take even and odd simple cells, and then square the output, and then essentially you're computing the length of this vector. I mean, this is really simple trigonometry like this, sine squared, cosine squared plus equals 1. So you remove the dependence on the phase, and that's the purpose of the V1 complex cells. But it seems that this minimum configuration is not the thing that's really implemented in V1. It's putting way over a larger area. And Kota did this study like this. So this is a simple cell model. And if you use these two and use even symmetric, odd symmetric, and squaring, and then you get a complex, a minimal complex cell. But the complex cells seem to do this. You have additional summing from this area, left-hand side and right-hand side, and then do this. And it's really going beyond what's minimally necessary for as complex cell function if you want to remove the phase dependence from its output. And that's the actual data. This is quota. And so minimal size of the complex cell receptive field energy unit is about this. And then the total size of the receptive field is larger. And that's the actual data. And this is from some time ago. And we did the binocular version of the study by these two people. I mean, the cat is looking at the space in front of it. This is a bird's eye view. And the actual receptive field of a complex cell, this is the actual data, reconstruction from the data, has a fairly large receptive field in depth. The z-axis is going that way. So yeah, the z-axis is that way. And it's fairly large, but if you actually compute the size of the minimal energy unit for this unit, it's quite small compared with the spatial extent of this. So it's good in that the disparity detector is organized this way, putting minimal energy unit across space so that it makes the cell disparity selective quite sharply, but it doesn't really care that much about the lateral shift of the stimulus. And it's useful for that kind of organization. But the point is that it's not a random pooling. It's very specific about its common maintaining the common preferred disparity. Okay. So summarizing what I have talked about, often why pooling is necessary. The answer often given is it gives you position invariance. For example, if you take a V4 neuron, which is selective to U shape, it shouldn't care about exact position as long as it's selective to U. So upper left corner and the bottom left and bottom right, and so on. All these individual position-specific detectors are pooled to produce position invariant U detector. And another reason that might be the case is subsampling, and will require anti-aliasing filter, so it needs some kind of low pass, which is essentially pooling. And by doing this kind of pooling, you gain robustness in exchange for spatial resolution. So those are the things that I think might be the reason. But before going on with this filter and then pooling dichotomy, I will show you some examples of high order filtering first. So let's look at this. I mean, this is what used to be called surround suppression. We call it the texture receptive field because it suits more to this purpose of this study section. Anyway, so we call it texture receptive field. So this explains the traditional notion of surround suppression. If you stimulate so-called classical receptive field, it responds well, but if you include the surround, it just suppressed by having the stimulus in the surround. But this kind of set is quite useful in detecting this boundary of having texture here on the left-hand side and having no texture on the right-hand side, about the same luminance and color. Okay, and we measure this by using this kind of stimuli. It's just a modulated sine wave by another sinusoid of different orientations and spatial frequencies. You can see the contrast is being wiped out periodically at the different orientations and frequencies. And from that, we can reconstruct the center surround organization of texture receptive field. Here, what's shown in red is the classical receptive field, and the bar going through it is the preferred orientation. So it's still selective to gradings or some oriented features. But if you stimulate along with the center outside, shown in blue, the area shown in blue, then the cell gets suppressed. So that's the suppressive area for these surround suppressive neurons. And so, I mean, we find that most of the neurons have this kind of, traditional luminance receptive field consisting of on and off subregions, but these are not. These are actually responsive to texture edges, the presence of texture on one side and the absence of texture on the other side, that will be the best stimuli for these neurons. And traditionally, people often thought that the CLF is surrounded by this donut-shaped suppressive region, but that's not really the case. This is very rare. Most of these suppressive regions tend to be organized in this manner, like Gabor functions or some of those variations. It's a little bit off. So I mean, we think that the suppressive, the texture boundaries also can be represented by this orientation-based, spatial frequency-based description. It's a little bit higher order feature than the luminance defined contours themselves, but you can use the same scheme. And this explains, what's the best stimuli for this type of neuron. This is just a leg portion of this, what's a zebra? Yeah, with horizontal periodic bars, but then here it's this stripe feature is absent, and that's the best kind of stimulus for this neuron, where the bars are horizontal, and then the no suppression coming from the surrounding left and right hand side of the leg. Okay. So the way to construct that kind of thing is by having complex cells organized in this manner, where the legs are present, you collect pluses, and then from the outside you produce a suppressive input. So it's like a positive input and negative input. So in a way, this fits my description of the filter that I outlined initially. Like you need to allow both positive weights and negative weights. But so I would say this is definitely a filter. And recently we've been doing, I guess it doesn't move, huh? Yeah. Binocular version of it, looking at the possible difference in the structure of center surround or the texture receptive field organization between the left and right eyes. And here is an example of where the texture receptive fields appear to be similar for both left and right eyes. But clearly for this one, it's very different. There is a strong suppressive area in the middle for the right eye, and that's no case for the left eye. So we think that this can allow texture boundaries to be used for stereo. Now, another example, this is really not my work, but it's been work by Simoncelli and Higa, and empty spectral receptive field, and I think this is a filter as well. So I mean, I'm asking a question here, empty neuron is simple or complex? I mean, I think it's basically simple cell. And the way things work is that if you have a translating motion, this motion, when looked at in the spectral domain, in the axis of fx and fy and ft vertically, temporal frequency vertically, and then two spatial frequencies horizontally, this motion's power is present on a plane that is going through the origin, the slanted plane in this frequency space. Okay, so I'm rotating this coordinate, but the rotation of the coordinate is just to show you that it's a plane going through the origin. So the model that was proposed for the MT neuron is like this. Higa, Simoncelli, Higa plane is here, and I think battery is dying here. Anyway, the V1 neurons are represented by those balls, and then the matching plane, you get multiple V1 input, only those lying for that on that plane, and that will give you the detection for that stimulus power. Maybe I have to wait a little bit before I turn it on. So I mean, here on the left-hand side, this is the V1 neurons' spectral receptive field. It's just a pair of balls. But so you will need quite many of the V1 input to make up a empty neuron, and that's their model. So it's a receptive field defined in the spectral space. It's a little, you need a little conceptual jump of defining in your head a receptive field not in the XY space, but you have to define it in the frequency space where the spatial frequency is omega x and omega y, and the vertical axis is the temporal frequency, how fast it flickers if you're looking at the single point. And so this one, I mean, is a Gabor. This three-dimensional spectral receptive field for one pair of balls is a motion like this. So it's a Gabor that is moving in one direction. So you need all of these, with different orientations of spatial frequencies to make up a receptive field like this for MT. Now, this is, I'm only showing the positive input, but MT neurons have been shown to have a negative input as well. This is from a recent work by Shinji Nishimoto and Jack Allen. And they've shown the positive excitatory input that is going through that is on the plane that goes through the origin in that fx, fy, ft space. These are cross sections, and this is the perspective view. But there is also a suppressive input into the MT neuron. So they're receiving both positive inputs and negative inputs, suppressive input. And combined, this best describes that structure. The central excitatory plane is going through the origin, but there are two side bands producing, contributing negative input. So, so these are two examples that I've shown in the areas going beyond V1. Actually, one case was V1, still V1, but V1's high order features. Now, in order to give you some possibilities about how you should think about pooling, I'm showing example by these two people's main work, where pooling happens in not XY domain. That's where you probably think of pooling when you think about pooling. Pooling detectors here and here and here, there to make the say receptive IT receptive field, for example, it's very large. I mean, in order to realize that kind of large receptive field with position invariance, you need to do pooling over space. But the pooling can be defined in other dimensions, like spatial frequency. And that's really obvious if you think about what the neural networks, those layers represent. Layers axes are not really just space. They also span spatial frequency and orientation and dimensions that are unknown to us, like we don't have labels for those dimensions. So I mean, you have to think of pooling in those dimensions as well. So this is just a stereo application. So I mean, in stereo, if you take left and right images and the point indicated by circles, sort of more or less corresponding regions, then the luminous profiles in those two left and right images are similar, just that they are laterally shifted from each other. And if you look at the luminance pattern and look at the spectral content, it's broadly, 1/f kind of distribution, but it's broadband essentially. But if you look at the V1 neurons tuning width about 1 octave, it only spans this wide. So you need multiple spatial frequency channels to encode all that information. But in order to get disparity information, I think you'd agree that it's better to integrate all these spatial frequency channel information. So there is probably a basis for, that kind of basis in thinking that this kind of spatial frequency channel integration takes place in the visual processing. So we note that if you decompose these into different frequency components, the feature of constant position shift remains across different frequencies. But if you look at this shift in the left and right sinusoid phase, in the phase domain, it's proportional to spatial frequency. That's a simple relationship that anyone will notice. So if you integrate across different spatial frequency channels, you need to take this into account, or the brain needs to take this account. So these are the two questions. Does this happen at all? And if it happens, do they integrate in a consistent way to, in other words, in the end, different spatial frequency channels may signal disparity, but if you want to create a neuron that is specific for particular disparity, then all channels should agree in the sense, if you integrate into a broadband thing, which integrates multiple spatial frequency channels, then the preferred disparity should agree across those channels. So that's the rationale for the experiment. If you don't do integration and rely on just a single channel, then the interaction between left and right spatial frequency input will be just circular like this. But if you integrate multiple spatial frequency channels, left and right channels are usually tuned to the same frequency. So if you integrate, it has to be organized this way along the diagonal, right? And we do experiments, reverse correlation. We just put up a sequence of left-right gradings optimized for preferred orientation. Just change the frequency combination in a random way, and also change the phase as well. So we pick 8 phases for each one. So there'll be a total of 8 phases for left and right gradings. And then we have different combinations of frequencies, each eye getting 11 to 13 different frequencies. So that's a lot. I mean, more than 10,000 total. And the reverse correlation allows us to do the estimation of the responses. It's actually a 4-dimensional data. We have left-right frequency combination. We're giving you a matrix element. But within that element, we change phase as well between the left and right eyes. So for each of the frequency combination, you can change left phase and right phase, 8 point and 8, total of 16 points. And that'll give you the phase tuning like this. So that's the entirety of the data. I mean, each of these tiny maps is the phase tuning map between left and right eyes. And then with that, each of these can be defined for different combinations of left and right spatial frequencies. So we did, because we're interested in just the interaction between left and right spatial frequencies, we just, you can broadly look at the luminance here in light colors here. So we just reduce the phase tuning into a single point. Whether there's a strong response, we put up a lighter color. And the map that's created is like this. It only is a map of left spatial frequency and right frequency. And you can see a clear diagonal feature indicating that this integration across different spatial frequencies happen in some of the V1 neurons. So the picture is like this. And we want to quantify how elongated those sub-regions are. And we just fit a ellipse onto that, long axis and short axis, and that'll give you the elongation. And we can define elongation index, and just a ratio of long axis and short axis of these angles. And if you summarize the data, it looks like this. Here we have the F1 F0 ratio. That's a standard index between classifying simple and complex cells. So simple cells tend to have a small elongation, and some of the complex cells really had long elongation. So is the disparity signaled across different spatial frequency channels consistent? And the answer is given by the next slide. we can analyze, we can create from that phase and frequency combination data a disparity tuning curve for this particular neuron. And we can even analyze the frequency band individually. So we separated the data into low frequency and high frequency, and separate disparity tuning are constructed, and you can see that the peak is aligned for the contribution from low spatial frequency and high frequency. So it really gives you an idea that it's pulling along the spatial frequency dimension, but it's done in a very specific way so that the disparity tuning is not canceled or destructively, destroyed by this integration process. And this kind of thing is quite nice. If you have, this is actual disparity tuning curve that is going up and down, but if you add enough spatial frequency channels, you can actually reduce these negative going side lobes and make the disparity tuning even sharper. Okay, so they had some consistent results that are consistent with our result. I mean, this is V1 result, but in V4, they showed that this negative going lobe is present less for neurons that are broadband. And that's their result from some time ago. So I'd like to summarize, I mean, why pooling? I mean, traditional notions, I mean, all these are probably true, but what used to be the reason that pooling is ignored by physiologists, that it's boring, may not really the case. I mean, it's quite striking that the pooling along the spatial frequency dimension can sharpen the disparity tuning, remove these false matches. As well as, it can improve disparity tuning. But I'm still left with these kind of questions. I mean, are they really all that different? I mean, having negative weights, I mean, we can miss negative input because it's hard to demonstrate inhibition or suppression in neurons, because suppressive input alone cannot do anything to a neuron, and we can't tell really the difference between zero response and so on. So I have to consider a possibility that maybe this kind of a connection might exist, that filter to filter connection without going through some pooling process. So that's my thought, and here are my colleagues that contributed to our lab's work. Thank you very much.
Thank you. Christian?
I'm trying to understand the model. Could we possibly go to your second slide where you had your first complex cell model?
Yeah. Second slide? I mean, the text slide or?
The one which had the first model for pooling simple cells into complex cells.
This one or not the?
I can't see it.
Oh, not on the screen. Sorry.
No, not the neocognitron. That right one. Okay. So what is this half squaring? Is it a synapse where you have to have at least 2 spikes from the earlier cell in a short integration period for
right.
another cell to fire?
Yeah, squaring part can be just some fluctuations of, say, firing threshold for firing. But I mean, the critical part is the half rectification. In other words, if the neuron is hyperpolarized, it doesn't produce any spikes. Therefore, all the negative input into the cell is essentially removed when the action potentials, when it's converted into action potentials. In other words, cell internally, as a membrane potential, can be driven both positively and negatively, but when it goes out, only the positive or depolarization will produce, fairly proportionally higher spike discharge. And in that process, it's if you actually, there have been some studies where this function is, power law function is actually measured, and the power law's exponent is close to 2. And why it's 2, there have been some arguments about.
No, so I understand why you'd want to, because that's
Right.
gives you the,
Yeah.
as you had later,
Right.
sine square plus cosine square gives you it gives you the energy out of it, and it gives you phase invariance.
Right.
But
yes.
I'm trying to understand how do you get it in synapses? Is it, as I said, is it that you have to get a pair of spikes from the earlier cell within a short enough period, and that leads to one spike on the output cell? Is that
No, I
on?
don't think input, EPSP integration really. It's the output function of what happens when you translate from membrane potential to spike discharge, and so I don't think you're talking about something like a coincident input.
Yeah.
Yeah, but I don't think
that's not it.
That's not it. I don't think.
So it's
The reason for this.
so it's just an accelerating nonlinearity.
Right,
in the
essential
membrane.
nonlinearity that at the output of every simple cell, I'd say.
Okay, fine.
Yeah.
Okay. One quick question.
So that's an extremely impressive result on the multiple frequency disparity agreement. So one thing I wonder is those cells that show that phenomenon near the top of that graph, do they have a range of disparities? Or are they concentrated near 0 disparity?
Well, it didn't cat, so we can't really tell whether it's 0 or not. We can talk about the symmetry of the disparity tuning curve. But it's, so
Okay.
Yeah.
Okay. Yeah. The other question was, I mean, potentially, I guess this kind of integration could relate to producing stronger signals for hard edges that are defined by multiple frequencies. Do you think that that is a reasonable interpretation of this data?
disparity defined edges.
Well, no, not disparity defined
okay,
edges,
okay.
but just
Right.
high contrast sharp edges,
Yes,
as
okay,
opposed
okay. Yeah, monocular edges. I mean, okay. Yes. Yeah, I think that's the reason. I mean, broadband stimuli in all the information about disparity can be used instead of just one octave band single cell tuning.
Okay. Yes, okay.
So I want to come back to your final thing about whether you could just pool the simple cells instead of having to do this kind of summing. So my impression is that a very large portion of complex cells are actually not perfectly phase invariant.
That's true.
So
yes.
Does that support your contention that maybe there isn't this perfect squaring and summing going on? Squaring and pooling going on?
Right.
Would that be more likely to come about in a model which did not have this pooling part?
I tend to think that that's probably a separate question. Most most V1 cells, for example, is a hybrid. It has a fairly clean, complex, ideal complex like structure like this energy model, but it has linear phase-dependent input as well. Just like Y cell in the retina is a hybrid of center surround linear component, and then Y cell also has this nonlinear portion where it seems to be tuned to high frequency texture. I mean, it's so I tend to think of V1's neurons, half-hearted. It's not a clear simple and complex cells. So it seems that it's a separate thing. I think we need to talk a lot offline.
Okay.
Is it okay? So, so in introduction, you said that in actual neurons that the processing doesn't change dramatically from one step to the other,
Yeah.
and I didn't hear any answer to why the neural structure is not gradually changing rather than changing step by step, unlike in the artificial neural network. So do you have any comments on that?
I don't have an answer to that. And what do you think? I mean, it's not really clear cut, and but it's true that as you go to higher visual areas, the features that they are selected to become really more complex than the lower order layers. And maybe if you employ some evolutionary process or learning process, I mean, these things might happen.
Okay, thanks. And so thanks to all speakers.